right, these are your unit notes for blood and blood spatter. Forensic serology is the study of blood applied to crime. There's many different branches of forensics. So for this unit, we're looking at forensic serology. Blood can provide various evidence to investigators at a crime scene. For example, blood spatter patterns can be used to reconstruct a crime scene and kind of show how an attack was made. It can connect an object with a crime. For example, if they find a discarded weapon. It can also connect a suspect with a crime. For example, if they find blood on a suspect's clothing. And it can also place a suspect at a crime scene. For example, if we find the suspect's blood at a crime scene, we can match it to them, and that way we know that they were present. All right, blood typing. Blood typing is one test we can do with blood, and it is considered class evidence, and it's really just good to rule out the suspects. It's very cheap, very quick to do blood typing, so that's usually the first thing they do, and we can eliminate any suspects that don't match whatever blood type we found. DNA profiling can also be done with blood, and that would be considered individual evidence because it can help to pinpoint a specific suspect. So blood can't easily fit into one particular category. It can be either one, depending on the types of tests that we run on the blood that we find. When we talk about blood typing is going to identify the presence or absence of particular proteins that are embedded in the cell. Like I said, this is a quicker and less expensive test than DNA profiling is, and it produces class evidence, but it can still be used to link a suspect to a crime scene, or it can exclude a suspect. So what blood typing is looking for is the presence or absence of cell surface proteins. That's what determines your blood type. So there are 8 different blood types. Kind of the main blood types are A, B, and O. A would have just surface A proteins. B has just surface B proteins. AB has both A and B surface proteins. And type O has none of the surface proteins. So A and B proteins called antigens are found on the surface of some red blood cells. So the antigens are the ones that are on your red blood cells. If a person's blood cells have the A antigen proteins, then they have type A blood. If the person's blood cells have B antigen proteins, they have type B blood. If they have both A and B antigen proteins, they have type AB blood. And if the person's blood cells have neither A nor B antigen proteins, they have type O blood. In addition to the ABO system, we also have what's called the RH factor. This is where we get those 4 additional blood types, so 8 total. About 85% of the population has an additional surface protein called RH factor, and we call them RH positive. The other 15% do not have that protein, and they're what we call RH negative. So again, just a different type of surface protein on those blood cells. Going back for a second, that means of those A, B, AB, and O, we can have AB positive, AB negative, depending on whether or not they have RH factor. We can have A positive or A negative, depending on whether or not they have RH factor, and so on. So that's how we get the 8 different blood types. To test for blood type, what we do is we take 4 drops of blood placed onto a well plate, and there's going to be 4 different wells that we'll use to test. And then we will add antibody serums to each drop, 1 type of serum for each of those different wells. So we'll add antibody A serum. That antibody A is going to test for any A antigens that would be on the cell. Antibody B serum is going to test for B antigens that are on the cells. Antibody RH serum is going to test for the RH antigens. And then we're also going to have a control well with no serum added. That way, if the control well were to react in some way, then we know that something went wrong and we would have to retest. Maybe, it was a contaminated sample. Maybe the well plate wasn't fully cleaned, whatever it may be. We would have to rerun that test because the control well should not react to anything. So once we add those serums, then we're going to watch those drops for what we call agglutination, which is similar to clotting. It looks like clotting to the naked eye. If it does agglutinate, that indicates that antigens are present on the cells. That means that the surface of those cells have those antigens present, right? The antibodies are going to attach to those antigens and cause it to agglutinate. So for example, this is kind of what we would look like. Here is what all of those different, every blood type, the test is going to be slightly different. If it does agglutinate, that means it does have that antigen. For example, O positive did not agglutinate for anti-A, did not agglutinate for anti-B, but did agglutinate for what's called anti-D, which is the same as anti-RH. So this means it does have the RH protein, but not the other two, which would make it O positive. All right, here's AB negative. It does have A antigens. It does have B antigens. It does not have RH. So that would make it AB negative. And again, here's our here's like an invalid test. You can see that the control, all the other controls are normal. This one agglutinated, so that means that this test is not valid. We would have to rerun that test. Here's what it would actually look like on a well plate. So when we talk about agglutination, we're talking about this clumping. The serum that's added to the blood contains antibodies. Those antibodies are going to attach to the antigens on the surface of the cell and cause them to clump together. If that antigen is not present, then the cells are not going to clump. So this one, if it does clump, that means that that antigen is on the surface. So this is clumped together. This is also clumped together. This one is not. So it does not have the B antigen, but it does have A and it does have RH. So that means it would be A positive. The other type of analysis we can do with blood is called blood spatter. Notice it's spatter, not splatter. There's no L. A grouping of blood stains constitutes a blood spatter pattern. The distribution, size, and shape of blood stains on a victim, on a suspect, or on the walls, floors, ceilings, or on objects can help us to reconstruct any type of event that would cause blood to be shed. Analysis of a blood spatter pattern can help to determine the direction blood traveled, the angle of impact, the point of origin of the blood, the velocity of the blood, and it can help us determine the manner of death. Blood stain pattern analysis can also help one evaluate the credibility of statements provided by a witness, a victim, or a suspect. The blood spatter should match a witness statement. If it doesn't, the blood spatter is telling the truth, whereas the witness may be bending the truth or lying. All right. Cohesion is a property that allows blood to stick together as it falls, maintaining sort of a rounded shape. Blood is going to resist flattening out when it falls on a flat surface. That cohesion and surface tension help to maintain this sort of curved shape, sort of like a bubble type shape, similar to like what water does. The shape of an individual drop of blood can provide important clues in an investigation. When blood falls from a height or at a very high velocity, it forms what we call satellite droplets, small secondary droplets around the main drop. So here is the main drop. And as it fell, if it's from a very high height or a very high velocity, we're going to get these little satellites around the edges. So the more satellites, the further the satellites are from the main drop, that's going to give us clues about where that blood fell from. When blood falls onto a less than smooth surface, the edges may have spikes or extensions. Spikes would be like the edges here, the edges and scallops of the main drop. Rougher surfaces are going to have more of those present than smooth surfaces. An elongated blood drop indicates blood was traveling from a different direction when it landed. So from the elongation, we can determine the point of origin and determine the angle at which it entered. So the point of impact may appear darker and wider than the rest of the drop, with a tail pointing in the direction of the blood's movement. So it's going to, the tail is going to point to where the blood came from. Smaller secondary droplets may break off and will land in front of the moving droplet, allowing scientists to determine the direction of the spatter. So you can see here, this is a pretty low angle. As it hits at that angle, some of that blood is going to spatter forward. When we see that spatter forward, we know that the blood came from the opposite side. The elongation of a blood drop varies based on the angle of impact. For example, 90-degree drops create a very round drop pattern with no elongation. Those are going to look like circles. High angles, 90, 80, 70 degrees, are all going to be very round, right? The lower the angle, the more elongation there's going to be. So you can see as we get into like 60 degrees, 40, 20, 10 degrees, it is more and more elongated, less round, more kind of oval shaped. So using the shape of the drop itself, we can at least determine if it was from, a straight up and down drop or if it was from a lower angle. All right, these are the main types of blood spatter that we will see. So there are passive drops. Passive drops are drops that fall at a 90-degree angle to the floor. They're circular drops. They may have secondary satellites depending on the height. Think about like if you have a bloody nose and you're walking across or walking down a hallway, if you're not stopping if you just let the blood fall to the floor, then you will make these passive drops. Right? Arterial gushes. This is when an artery is severed or injured and is like spurting blood. So the blood pressure from severed arteries results in gushes of high blood volume. So we'll have like very high volumes of blood, so much so that it starts dripping. So these drips came after the blood was deposited by the artery. We have what are splashes. Splashes are similar to passive drops, but splashes refer to having different angles. Passive drops are from 90 degrees. Splashes are going to be from lower angles. That's going to help us determine the position of the victim, the direction in which the blood was moving. Then we have smears. Smears is when like a bleeding victim would touch the walls or furniture. So if they're, holding an injury and then lean up against something, they'll leave a smear of blood. A trail shows a victim moving from one place to another. So whoever was bleeding, you can follow their path of movement based on those trails. They may or may not be passive drops. They could be passive drops. They could also be arterial gushes. It's going to help us determine the direction that the victim moved. And then pools are from when victims bleed very heavily and lose a large amount of blood in a very short period of time. The blood is going to, pool in one area and become rather deep, depending on the amount of blood that is lost. So not too specific of a pattern here. It's more about the quantity of blood when we look at a pool. All right, speed and velocity affect blood spatter. Generally, the higher the energy, the greater the division and the smaller the spatter. Generally speaking, the lower the energy, the less the division and the larger the spatter. So high velocity, small spatter, low velocity, large spatter. Let's look at some examples of that. So for example, high velocity example would be a gunshot wound. This is a very small diameter, very high, powerful, quick injury. You can see that the individual spatters, right, the individual, what we would call maybe drops of blood are very small, right? It's kind of a very fine spray of blood. That's what we mean by the larger or smaller spatter. We don't necessarily mean the area that it's taking up. We're talking about the individual places that blood can be found are very small. Medium velocity would be something like a beating or a stabbing, talking about 1 to 4 millimeters for the blood. So you can see here it's slightly larger than what a gunshot wound is. Gunshot wound is going to look just like very, very fine, less than 1 millimeter drops, whereas medium velocity is going to be 1 to 4 millimeters. Low velocity would be a blunt object impact. Here's like a hammer, for example, a very large spatter pattern from a very low velocity. Then we have something called cast off. So the movement and the number of swings can often be documented by examining the cast off pattern, what we call a cast off pattern. During a beating with an instrument which produces the bleeding, blood will not normally collect on the surface of the instrument from the first strike. On subsequent strikes at the same location, blood will adhere to the instrument since it now strikes a blood source. When the instrument is raised or swung backward, its movement allows small drops of blood to be released from its surface. So cast off is the blood left behind from the weapon. So that very first initial hit, there's no blood on the weapon yet. After that first initial hit, every time that weapon is drawn back, it's going to leave blood along a wall or a ceiling or something like that. And that's what we call the cast off, right? As they bring the weapon backward, we will get that cast off stain showing that the weapon is moving backward. Additional subsequent hits are going to provide more and more cast off. So for example, here, this would be the trail from one swing This is the trail from a second swing. We can determine how many times the weapon was struck. So this is the pullback after swing 1. This is the pullback after swing 2. So there could have also been a third swing. Right. So it looks kind of like trails of droplets, and based on the elongation, we can determine the direction that the weapon was struck. So when we find blood evidence at a crime scene, blood evidence is not always evidence. So even if we don't see blood, but perhaps, believe blood could have been there, even with the most thorough cleaning, blood is going to leave residue that's very difficult to remove. Forensic scientists can use something called luminol powder. They mix it with hydrogen peroxide and spray it over an area, and that luminol is going to illuminate any leftover hemoglobin left behind by blood. You can't see the hemoglobin. It's like a microscopic molecule, but that's what the luminol is going to bind to and react with in order to create that light. And it's going to luminesce for about 30 seconds. So it's not permanent, not permanently lit up. It's just going to confirm whether or not blood was there. So you can see here, no blood. Once we spray it with luminol and shine like a black light over it, then it will glow. So even though it looks clean, there is blood residue there. The luminol reacts with old or new blood. However, it destroys the blood, so it can't be tested later. That's why we're going to just do like one test with luminol. And then like on this carpet, for example, if we do see blood residue, we're going to collect that piece of carpet, see if we can get any blood from the underside of the carpet or the padding of the carpet. Anything that was attached to the luminol is going to be destroyed. But if there's blood that did not react with the luminol, we'll be able to still test that. Next, if we do see blood, we can't just automatically collect it and test for blood type. First, we need to determine if it's actually blood, right? There are ways to make fake blood. Think about like Halloween costumes that have fake blood. We need to make sure it's actually blood so we don't waste resources. There's a lot of different ways we can test for that, but one of the most common tests is called the Kastle-Meyer test. They basically take a swab that is soaked in a solution, and they just, rub a very small portion of the blood spatter. If it turns pink, that means yes, it's blood. If it does not turn pink, no, it is not blood. Very simple. That's a test that can be done very quickly. Only takes a couple of seconds, so that we know if we need to collect that blood for testing for blood type or DNA. Once we determine that it is blood, not always, but many times we need to determine if it is actually human blood. For example, if a crime takes place on a farm, we need to make sure that it is human blood and not blood from, a cow or a pig or something like that. There's a test called an ELISA test that uses antibodies for many different organisms, and it will react with human blood, and it will be able to distinguish other mammal blood from human blood. In rare cases, it can be confused with chimpanzee or gorilla blood just because we're so closely related to those organisms. However, the likelihood of it being chimpanzee or gorilla blood is obviously very low. But that test again is expensive, so it's only done in cases where we suspect it may not be human blood. If the blood is human, we're going to try to determine whose blood it is. The first thing we're going to do is test for blood type. Again, it's cheap, it's quick. If we need more information, if we need that individual information, then we're going to do DNA analysis as well. So we're not going to use all the blood for blood typing. We're going to make sure we save some if we need to do DNA analysis later. All right, those are the end of your notes for Blood and Blood Spatter. Please make sure you submit them on Canvas.